The year is 2019, and we were promised marvelous innovations in almost every aspect of life. However, we still drive our own cars, clean up our own messes, and are rapidly being displaced by AI. Our heroic trio of disruption students, with the help of our all-knowing professor, Big Brother not included, is trying everything they can to complete the deliverables for our project.
So you all have a lot of deliverables you'll need to complete by the beginning of next week, which you all now know is on Sunday. Remember, there are a few directions and I have offered no further guidance whatsoever. If you need to ask questions, I promise to answer them with the least amount of sarcastic undertones as I possibly can.
How are we going to know what he actually wants?
Well, it's almost like we have to turn ourselves into Shannon.
It's a great idea. We should do that.
Are you being serious?
As serious as this deliverable.
Well, how can we make ourselves more like him?
Well, maybe we can take one of these gene therapy pills I'm reading about.
Let me see. It really only seems like they have ones that prevent or cure diseases, but what we really need is to help with our thinking abilities.
And I don't think I have $250,000 laying around for this anyway.
First of all, that is just way too expensive.
It's basically a form of price discrimination.
We're definitely going to have to go with something cheaper.
I think I have an idea of something we can do. It's a lot cheaper, and the Chinese have already used it to make 2 of their babies smart already.
What is it?
A DIY CRISPR, of course.
Let's do it.
I mean, I guess.
Welcome to the world of the infinitely small. From blood cells to mitochondria to quarks. Every single person contains a genome. What is a genome, you may ask? A genome is all of your DNA that contains the information necessary to build and maintain the organism. DNA stands for deoxyribonucleic acid, which is a series of instructions. The structure of DNA is a double helix, and in that double helix are 4 chemicals that are building blocks: adenine, thymine, cytosine, and guanine. Adenine binds with thymine, and guanine binds with cytosine. All 4 of these building blocks are read like a code, which are 99.9% similar in all humans. Yet, those few variations in our genetic code contribute to our differences. In order to figure out where our individual differences come from, scientists can use DNA sequencing to determine the order of the 4 chemical building blocks, adenine, thymine, cytosine, and guanine, that make up the DNA. By understanding this pairing, scientists can effectively sequence the next pair in the process. The human genome contains approximately 3 billion base pairs, so there is definitely a lot to unpack. You got here quick.
Nice.
Alright,
Let's do it.
let's see what we got here. So, got this.
If we want to become more like Professor Shannon, I think we should start by removing our genes that enable empathy for people that turn in things late, of course. Next, we need to add some wit, definitely some gray hair, and some deadpan sarcastic humor. And then after that, we just got to make ourselves really smart.
I just have a question. Is the gray hair super necessary?
Absolutely.
Definitely.
CRISPR stands for clustered, regularly interspaced, short palindromic repeats, and its associated protein CAS9 act as a pair of molecular scissors, which allow scientists to remove or change an organism's DNA. So, how does CRISPR work? CRISPR was originally adapted from a naturally occurring genome editing system in bacteria, where the bacteria captures pieces of DNA from invading viruses and uses them to create DNA segments called CRISPR arrays. These CRISPR arrays allow the bacteria to remember the virus if it attacks again, where it will produce RNA segments from the CRISPR arrays to target the virus's DNA. The bacteria then uses CAS9 to cut the DNA apart, which will ultimately disable the virus. This is how to visualize the process.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Does any of this sound too good to be true? This could be used to actively reshape medicine if used correctly. However, this procedure is not recommended or outright banned in some countries, and germline, heritable, gene editing should not be used for reproductive purposes until it has been deemed completely safe. Most of the genes edited are somatic cells, which are not sperm or egg cells, but rather cells that only affect certain tissues and are not passed from one generation to the next. It is worth noting that this technology is still in its relative infancy, and it is important to approach the idea of enhancement with ethical considerations in mind.
Yeah, so I was thinking more about this CRISPR thing, and I'm definitely having some second thoughts.
Yeah, me too. It's just like the DIY gene editing thing really does sound risky when we don't know what the consequences are.
You're right. What if we had some sort of limitless pill that would boost our intelligence?
I like that idea, but how would we make a limitless pill?
Oh, wait! We could 3D print one!
So what is printable medicine? Printable medicine is a new way of delivering medicine to a patient. This allows doctors to print the medication in their office and allow them to administer more than one medication in one dose, and it's completely safe for the patient. So, how is printable medicine made? The technique was inspired by organic vapor jet printing. First, a powder version of the active pharmaceutical ingredient in it has to be heated until it evaporates. Then, it is combined with an inert gas, and an example of this would be nitrogen. Lastly, the mixture is funneled and sprayed onto a cold surface so that it crystallizes and becomes a thin film. This technique has made it easier for doctors to layer up on medication for a patient, and this way, it's easier for the medicine to dissolve into the patient's body. There is no need to add any other chemicals because the pure powdered form of what will help the patient is all that is needed. Like a lot of rapidly advancing technologies in this space, there is a concerning lack of regulation. How do you know that the medicine that you've 3D printed is actually going to work? What if the drug you're given is little more than a placebo? This is one of the challenges that lawmakers are going to have to address going forward.
Wow, this 3D printer is pretty incredible.
It really is.
It's really awesome that we can 3D print medicine right here on campus.
What ingredients do you need for that again?
A lot of chemicals and placebo. Pardon me, but I think you're in the wrong place.
Wait, who said that?
I did. I hacked into your computer because your password is password123. In addition, your webcam is uncovered and there's nothing in the audio jack, so I can hear what you're saying. Oh God. You can 3D print a massive variety of complex models, such as tissues with blood vessels, bone, heart valves, cranium replacements, organs, and medicine. But what you're trying to print requires ingredients not found at the university. However, there is a major difference between 3D printers and bioprinters. 3D printers will ultimately print something that is composed out of plastic or metal, as opposed to a liquid or a gel-based material. Also, in the future, artificial intelligence and machine learning are going to be playing a big part in biotechnology. The first implication is in diagnostics. Machine learning will be able to find a commonality in diseases and will be able to detect it faster. As the test is done over and over again, the more it will learn about what to look for and even mutations of diseases. You're going to want to look into developing a biointerface. In its simplest form, a biointerface is when DNA, RNA, an antibody or protein comes in contact with any surface. In more advanced applications, biointerfaces typically include more complex biological organisms such as cells, membranes, or viruses, which are connected with synthetic materials. The advancements in biointerfacing, biotechnology, and genome editing have the potential to drastically change the future of work for all human beings. Medical advancements typically increase life expectancy and quality by finding cures for life-threatening diseases and augmenting human capabilities to counteract health problems that lead to the inability to be active and work. This is definitely the case when in regards to innovations that are currently in development or that have just recently been rolled out to the public. Innovations like neuroengineered biointerfaces, individually designed prescription cocktails, and the editing of one's own DNA will likely expand one's life expectancy and therefore prolonging their ability to work by decades. Thanks for listening. Please make smarter decisions as far as your security is concerned so that no one else can hack into your computer.
So 3D printing didn't work.
Well, Shannon did mention something about those Alexa sensors the other day, and I actually picked one up and I have one in my pocket right now.
Oh my god.
That sounds great. I mean, I guess it would give me the answers to everything, but how would you insert it?
This stapler should do the trick.
Oh my god, I can't look.
Oh, God. Are you sure this is going to help us be more like Shannon?
Absolutely. Did you want me to explain it again?
Sure.
All right. So a biointerface is when a DNA attaches to a surface, which in this case is our Alexa chip.
Okay, so I get this Alexa chip and I'm going to have access to basically an infinite archive of information. So I'd say that's kind of like Shannon.
That's exactly right. All right, here goes nothing. Fingers crossed. Wait, what?
Who's that? Hello?
Hi, Max.
Mark Zuckerberg?
Yeah.
How did you get my number? Why are you calling me?
I totally did not look through your Facebook account and find your location and look into your recent cookies to see why you were looking up how to insert biointerfaces without hitting a major artery. But that is sort of the main reason why I thought it was so important to get in touch with you before it was too late. Well, you see, I've gotten myself into some trouble with keeping people's data private.
So, how are those trials working out for you? And I hear that you buy up all the property that's near where you live.
Oh, damn.
He really values his privacy.
Stop laughing or I'll make sure you'll get spammed with very convincing pyramid schemes. Tummy tea, anyone? So, Max, an implantable system, like that chip, would be used as a sensor or as a monitor. Say, if a person has a bad heart condition, that sensor would let that person know when their heart is beating irregularly and when they should seek medical attention. However, since it can tell that person with a notification that their heart is acting abnormally, who says that it can't inform that person's employer or their insurance company? An individual's health database that is hypothetically available to an employer during an employee selection process could have very negative consequences if they have history of poor health or of drug and alcohol abuse, all of which could be recorded if there was an insertable biointerface used. This could potentially lead to discrimination during a hiring process or even during actual employment. Yeah. They say those types of devices aren't hackable, but guess what? People do lie. And it is very easy and compelling to sell a person's private data to a third party to make even more cash than you could ever imagine. We definitely have not come that far in that technology to be able to use an insertable chip to increase someone's intelligence. And honestly, when we are able to create an implantable with those types of abilities, the cost will be so high that a person with an average income will never be able to afford such a thing. If the chip is inserted wrong, the tissues will reject it, and all that precious time and money you spent will go right down the drain.
That's pretty weird. I need to get more secure.
You know, I don't trust him. He talks a lot of game about privacy, but like, he won't come out and say that he's selling our data.
He did make some pretty good points about the purpose of biointerfaces, though. And to be honest, I'm not entirely sure the stapler idea I had is actually going to work.
Yeah, I agree.
He even really said it himself that technology hasn't developed yet, though.
So now what do we do?
Well, here's a video of what biointerfaces are actually supposed to do. It's pretty cool.
It's tough to deliver medication quickly and precisely to different parts of the eye. Traditionally, medicine for conditions like glaucoma or diabetic macular edema is injected into the eye or dropped onto the surface and left to diffuse slowly throughout. This delivery method isn't always accurate and means the entire eye receives the drug. The process can be slow, irritating, and painful for the patient. Researchers wanted to build tiny delivery vehicles they could drive to deliver cargo quickly to specific parts of the eye, particularly through the dense jelly called the vitreous humor. So they used nanoscale 3D printing to create small, spiral-shaped delivery bots. The bots were nearly 200 times smaller than the diameter of a human hair. Their tiny size and a non-stick coating made it easier for them to pass through the tight matrix of the vitreous humor. The spiral bots also contained magnetic material, which meant the researchers could drive them forward, backward, left, or right with a magnetic field. The scientists injected a solution containing around 10,000 of the spiral bots into eyes collected from pigs and put the eyes in a magnetic field. They used imaging techniques in dissection to see if the bots had been able to pass through the eyeball's jelly all the way to the back where the light-sensing retina lives. The bots were able to deliver their cargo to the right place in much less time than simple diffusion would allow. Next, the researchers are planning to test the spiral bots in a living animal's eye. It will be years before any humans have tiny spiral robots delivering drugs to the back of their eyes. But the work is a step forward in building controllable vehicles to aim medicine at precise targets in sensitive areas of the body.
All right, so what should we do about this now?
Probably go back to the house and do this deliverable without Shannon's all-knowing knowledge.
All right, well, we'll see how that goes.
Yeah, really. Well, that was a major bust.
You ain't kidding. Have we checked that CRISPR kit in a while?
What are those little white dots in the petri dish?
Oh my god, it worked.
There's no way that thing just worked.
Wow, that's crazy.
Let's shoot it up. Nah,